Hello, it's me, Oscar from Marijuana Garden.
Imagine you're given a string of beads, hundreds, even thousands of them. Each bead is one of 20 different types, each with its own tiny magnetic or sticky properties. Your task? Predict instantly the incredibly complex, unique three-dimensional shape this string will twist and fold itself into. This shape determines everything the bead string can do. That's the protein folding problem, arguably biology's grandest challenge for over 50 years. Proteins are these bead strings, chains of amino acids. Their folded three-dimensional structure dictates if they become an enzyme digesting your food, an antibody fighting infection, a muscle fiber contracting, or the spike protein a virus uses to invade cells. Knowing a protein's structure is the key to understanding life, fighting disease, and designing new solutions. For decades, scientists like David Baker, a computational biologist at the University of Washington, were protein detectives. They used powerful computers and complex physics simulations, his project was called Rosetta, to try and predict structures. It was slow, required immense computing power, often borrowed from volunteers' idle PCs via Rosetta at home, and even turned into a game, Fold It. Still, it was only accurate for small, simple proteins. Figuring out one structure could take years and millions of dollars using lab techniques like X-ray crystallography. Millions of known protein sequences had unknown structures, a massive roadblock for biology and medicine. Meanwhile, Demis Hassabis, a neuroscientist, AI researcher and CEO of DeepMind, an AI company owned by Google, and John Jumper, a brilliant computational scientist leading the project at DeepMind, had a bold idea. What if we could train an artificial intelligence, specifically a deep learning system, on the tens of thousands of protein structures already painstakingly determined in labs? Could the AI learn the hidden rules of how an amino acid sequence dictates its final, three-dimensional shape? In 2018, DeepMind's AlphaFold entered a prestigious global competition called CASP, the Critical Assessment of Structure Prediction. Its performance was impressive, but not revolutionary. Then came AlphaFold 2 in 2020. What AlphaFold 2 did differently was pretty amazing. First, instead of relying only on physics simulations, AlphaFold 2 was trained on a massive database of known protein structures and sequences. It learned patterns humans couldn't see. Second, it analyzed hundreds of millions of related protein sequences from different species. If two amino acids in a protein consistently changed together across evolution, it suggested they were probably close neighbors in the three-dimensional structure, a crucial clue. Third, AlphaFold 2 used what's called the attention mechanism. Think of this like the AI focusing a powerful spotlight on different pairs of amino acids in the chain, asking, how likely are these two to interact, and how does that affect the overall fold? It did this millions of times simultaneously. Finally, it didn't just predict contacts, it built a highly accurate three-dimensional model, including the precise angles of the bonds. Designing drugs is, well, a lot like making a key for a lock. The protein target. If you know the lock's exact three-dimensional shape or structure, then making the right key or drug becomes infinitely faster and cheaper. This, honestly, accelerates cures for cancer, Alzheimer's, infectious diseases, and rare genetic disorders. Many diseases like cystic fibrosis and sickle cell anemia are caused by misfolded proteins. Now, we can actually see exactly what's wrong at the atomic level. And revolutionizing biotechnology and sustainability, this is your future. Want an enzyme that eats plastic pollution? Or maybe one that breaks down plant waste efficiently for biofuels? Or even captures carbon straight from the air? Engineers are now using these AI tools like Baker's RF diffusion to design proteins from scratch with specific functions, creating new materials, clean energy solutions, and sustainable industrial processes. This is synthetic biology on steroids. From improving crops to understanding cellular processes, knowing protein structures removes a massive bottleneck and really accelerates all biological research. Grad students and researchers worldwide now start their projects with predicted structures. While they haven't won the official Nobel Prize yet, science prizes move deliberately, their achievement earned the 2023 Breakthrough Prize in Life Sciences, which is often considered the Oscars of science, and a strong indicator of Nobel worthiness. The sheer scale of the problem solved and the transformative impact on all of biology and medicine is undeniable. As a college student, this revolution is happening during your lifetime, and honestly, it directly impacts your future. This is biology, computer science, chemistry, physics, and AI all colliding, an incredible interdisciplinary opportunity. No matter your major, skills in data analysis, computational thinking, or biological understanding are crucial. The toolbox is open. The AlphaFold database and tools like RosettaFold are largely free and accessible. Future researchers, maybe even you, will build upon this foundation. The ability to understand and design proteins is key to tackling climate change, with carbon capture enzymes fighting pollution, with plastic-eating enzymes supporting sustainable food production and developing next-generation medicines. You will be applying these tools and be inspired. This shows how fundamental curiosity-driven research, combined with cutting-edge technology like AI and open collaboration, can solve impossible problems and benefit all humanity. It's honestly a blueprint for future scientific breakthroughs. Baker, Hasabi's, and Jumper, along with their teams, created AI systems that can instantly and accurately predict the three-dimensional shape of almost any protein just from its amino acid sequence. This unlocked the instruction manual for the molecular machines of life, accelerating medical breakthroughs and empowering us to design new biological solutions for a healthier, more sustainable future. It's a foundational leap for biology that empowers your generation to build a better world.